Okay, so this will be a talk on the history of exon skipping technology for the treatment of Duchenne muscular dystrophy. So we begin with Dunkley's paper, Dunkley and others' paper, and they hypothesize that dystrophin expression in MDX mice can be restored to normal levels if we are able to skip exon 23 of the murine DMD gene. And the reason why exon 23 leads to some issues is that there is a nonsense mutation within this exon that causes a premature halting of the translation of this gene. And what Dunkley is proposing is binding this antisense oligoribonucleotide to the intron, the 3' splice site between intron 22 and exon 23. And what we'll see is that he found that if such a process, such transfection occurs, then the sarcolemma of about 1% of MDX myotubes show evidence of dystrophin expression. And by the way, the way we show, or the way Dunkley showed that dystrophin was expressed is through fixing and staining transfected myotube cultures with this C5 monoclonal antibody to the C terminus of dystrophin. And so here's an image that demonstrates the difference between dystrophin expression. So in B, that we see that there is dystrophin expression in this, the approximately 1% of cultures described before. In A, this is the majority of the cells, majority of the cultures, and you see that there is no dystrophin expression at the sarcolemma. So Dunkley was able to show, although this is not a very efficient method, that complementary RNA to 3' splicing site of intron 22 can skip exon 23 and hence induce dystrophin expression. Now Gebski improved on this by introducing this idea of binding the AO to what is called a leash, which is kind of like, we'll see here, is kind of like a complementary segment to your AO. And these are the 12 examples, but let's go back. And what Gebski showed is that not only does it induce exon 23 skipping, but it does it in a more efficient manner than previously done by Dunkley. Okay. So how did Gebski measure this efficiency? Firstly, he checked or tracked the delivery of this morpholino AO bound to leash 4, and if there was fluorescence exhibited in the culture, then we could say that it was delivered. In this example, if we take a look at D, this is this AO with the leash 4 that I tried to mention earlier, and we see that there is this fluorescence, so we can see that it's been delivered. And in the other cases, these are the controls that did not show the delivery. Gebski and others showed that exon skipping was in fact present, was this analyzing the presence and relative intensity of a 688 base pair RT-PCR product, which characterized the missing the mRNA corresponding with the missing exon 23. Gebsky's main result is that morpholino AO is effective at skipping exon 23 at nanomolar concentrations when annealed to a leash duplex in complex with this lipofection. Yeah, and then the conclusions much of the same.